Thanks, Jeanne. Eric, David, would you like to join us back up here, and we'll go through a panel. That's some really fantastic data, Jeanne, and as we discussed kind of in some of the pre-conference discussions, one of the things that you were discussing was the fact that, you know, are other labs doing the same kind of work? Are they really investigating at the level to create assays, to be able to characterize to this level?
So there are examples. I mean, I go to a lot of conferences. I went to the International Stem Cell meeting in San Francisco, ISSCR, just this summer. And I went to a clinical translational program, which was at the beginning of the conference. And I couldn't believe how many people stood up and said, okay, we're working with the FDA, we karyotype the cells. And I thought, why don't you do something more than karyotype the cells? And they would say, and they said, two of them said, that's all the FDA requires. And so that's, you know, that's essentially all I can do right now. I could lead by example. I can explain to people that none of this stuff is all that hard, and it's going to save us a lot of trouble downstream. But that doesn't mean that people are going to abide by it, because ultimately they don't want to invest in anything that the FDA doesn't require.
I think part of the challenge here as well is it's probably absolutely fair that that's all the FDA want to see, but to prove what? Mm-hmm. And I think that because people in basic research on the whole have never previously had to engage with regulation, then they wouldn't understand the difference between, you know, developing a therapeutic or a laboratory-developed test. So consequently, I think that to support all the efforts which you're going through, I think there's a need to integrate some basic product development into education. Whether that's your PhD program or master's program or whatever, it should be a part of it, because it's the narrative thread of product development.
Yeah, I agree. And I think Thermo is probably going to be a much bigger player in that space. Yeah. Don't you think, Eric?
I do indeed, Jeanne.
Eric is going to teach everybody.
Yeah, that's what I asked him to do, and he said sure.
I think what you're doing is fantastic, right? To be able to have more information and understand what your cells are doing. I mean, it's all about patient safety here,
yes,
right? I mean,
exactly.
we're trying to develop technologies that are going to help people. We don't want to make them more ill, of course. So the more information we have, the better we can make choices about the technologies that are going to be necessary to move these things into commercialization. So that's a great thing. There always is the balance of what you need to do from a regulatory perspective and what you should do from a development and research perspective. So that balance always has to be met. And what happens often is that you will develop the technologies and validate towards more simple indicators potentially that will be used in your commercial manufacturing, but you have to have that underlying information and data set to be able to make those correlative indications for assays that you can use for release testing. So that's
yeah, and that was the idea behind Pluritest. Was you can still get a lot of information. It costs $150 to do a gene expression array, and you don't have to know anything about bioinformatics. You just upload the data. So that has led to adoption of that kind of assay, and it's made people who've never done any genomics at all feel comfortable in using a test like that. So all along the way, we're trying to make things easier for people to do. I mean, analyzing whole genomes is never going to be really easy, but there's so many people who can do it now. You know, it's no longer the purview of, you know, like the 5 greatest labs. There are hundreds of places that can do really good genome analysis. So that doesn't need to be a barrier even. And it's also cheaper now. It costs, well, it theoretically costs $1,000 to sequence a genome. It actually costs like $1,500. If you can talk people down, it costs about $1,500. And then you have the data, and then you always have the data. So it's remarkably, everything has evolved, I think, in that direction to make these things easier. And a lot of it has to do with companies like yours and other companies that do sequencing and other assays.
Yeah, the information out there is it's incredible, and being able to tie it all together to one of the early themes of this talk is big data. You know, the information is there, and how do you track it? There's incredible amounts of it, and it relates all the way to patient delivery, understanding, you know, which patient might be.
I agree. Yeah. We're sequencing the patients too,
Right.
you know. I don't know what that's going to mean, but if something happens, we have something to go back on and try to figure out why it happened.
Exactly.
I think it's also important to be positive about this, because it's not that people aren't working with the FDA because they're being deliberately awkward. It's that the new therapies we have and the new tools we have, we simply haven't used before in such volume. So it's not that the momentum isn't growing, it's just that our expectation is possibly, and that of patients in the market, is possibly outstripping our ability to deliver on that. And I think it's always good, as you did excellent in your presentation, to take stock and say, well, what have we learned? And please don't make the same mistakes. I've made them for you.
Great.
Right, and I also think that there were technologies out there like mass spec, flow cytometry, where, you know, years ago it had to be an absolute specialist team within a university handling these things. Now with kind of the tools and technologies companies out there making these things more accessible, making the data comprehension of that. That is the one thing I was going to ask you. Do you see kind of that same bioinformatics interaction growing at Scripps, at other universities, at Oxford?
Yeah, so I decided a long time ago that I was going to do everything in-house. So, because I've had, you know, even though I have lots of collaborators, I always wanted to be, I wanted to be able to control the experiment so that I wouldn't have variables based on people's personalities, you know, that sort of thing. So we still do all our bioinformatics in-house, but as I said, I also collaborate with the Venter Institute. Those guys are really good. And I'd be happy to expand this more. It's just that it has to be, I have to trust them. And it's like trust that I have with reagents that I get from Thermo. I can't analyze them myself, and so there has to be a level of trust. And I think especially with big data, you need to, if you don't understand how to analyze it yourself, you have to trust the person who's doing it. And there doesn't seem to be a way of validating.
So I think there's sort of two approaches to that question. So one is the generation of the raw data, which
Right.
Jeanne mentioned. But the second thing is then the analysis of that data. So I think even though you very fairly said there's now hundreds of labs that can do, you know, genomic sequencing to a high standard, that's probably still fewer in number than the number of groups that potentially could analyze that data.
Yes.
So to your question, you know, about Oxford, I think Oxford has taken the approach that because Lucky Shing, the shipping magnate, gave them tons of money, they built a big data center, but actually they're using it to interpret other people's data sets. I think there's definitely a need to ensure that there's consistency in the standards of data collection and data analysis. And I don't actually think in genomics we're even at a point where the data collection is sufficiently standardized yet, let alone the data analysis on top of that. And I think that if we look further downstream, and Jeanne, I think very fairly but provocatively said, well, if we look at individual patients, what does that mean? Well, if we're using that to make clinical decisions, then there's got to be a very robust way of validating that. I think, Jeanne, I take you up on your offer of collaborating. Let's talk about that later.
Absolutely.
Yeah, and I think also one of the things I was going to ask was if you look at some of these kind of expedited pathways, is this data, I know you're saying you're talking to the FDA and hoping that's kind of going to push along your clinical trial and your approval to get started. You know, are there ways, David, within regulation or within these expedited pathways that are accepting or are more open to if there is more data in those filings? Is that one of the
So I guess I'll give the academic answer, and then the, you know, because I'm a road between two thorns with you and Eric here, the commercial answer. I think the academic answer is that, you know, almost all data is good data, and the more we understand the richer the data set, the more conclusions we can draw. I think the challenge when we look at an industrial setting is, well, once you have that data, you must interpret it in some way in a regulatory submission, and the more data you generate today is more data to analyse from a pharmacovigilance viewpoint moving forwards. And sometimes you don't necessarily want to do too much pharmacovigilance because you might find things you don't want. So I think that it's very important to define what is the purpose of collecting this data. And I do think that as this blur appears between what is a trial and what is a product which has got a conditional authorization, there should be a church and state between this is development and this is product adoption and a clinical issue. Because if not, we're simply going to have people paranoid about doing further development. And that's going to stymie the development of the product in the adaptive pathway, but also secondary products coming down the pipeline.
Yeah, so my goal is to find out all that stuff that I don't want to know.
Yeah, exactly.
But I think, in a weird way, I said, perhaps sort of too jovially, they don't want to know.
No,
Actually, we all want to know to reduce the risk.
Right.
The problem is that to that individual asset, it could be the death of it. So,
I know,
so I think that for large pharma companies, actually, it's not too much of an issue. That risk can be managed in a portfolio approach. But if you are, a Kite, a Spark, a Juno, a Bluebird, all the companies which Eric and myself mentioned, which only have 3 or 4, that could be really, really quite challenging. So I do think a church and a state is required. And I guess the analogy I'd use is, imagine that what you essentially have is 2 types of prototype. You have one which is a prototype which will support a future clinical product. And then you have a prototype today that is a living clinical product, and everybody is aware of the risks it has moving forwards. But I really don't think that having, unless there's obviously huge clinical issues found, I don't think there's a huge amount of value in taking conditional approval, supporting somebody to get more data, and then taking it off the market. Because there's a balance between actually what can you learn out in the market itself.
Right.
Right, but at the same point in time, I think the thing that Eric and I come across very often is people that don't have as much information as Jeanne has, but start with a very manual process like we discussed, and then how do you shift platforms if you don't have comparability? So Eric, I don't know if you kind of have comments as to the things you've done.
Well, I think these are really interesting questions that we're asking, and I think the technology now is expanding to a rate where we're going to see, almost a Moore's law effect in biology, right? So the Human Genome Project and now the different technologies for doing next-gen sequencing are so readily available, and only $1,000, and you get your answers in what, 6 hours, 8 hours, I don't know, whatever it might be. That's just the beginning. So now we have the ability to capture that data. Now what to do with the data is the next step, and then how to validate that data and start tracking. We don't have the answers today. And to the point of what is risk versus not, knowing what there is and then doing a risk assessment later on, or perhaps, understanding or finding out more information later from a clinical population, it's going to have to happen at some point. And the more information we have to go back to, which is one of the points that you were just making, the easier it will be to understand what may or may not have gone wrong. For example, in a serious adverse event or adverse event in some
I think we also need to go from this culture of change control, implying it's a bad thing, to kind of change management, because actually the amount of times we're going to have to do change control in a process is just increasing exponentially.
It's really hard believe me, that's the hardest part when you're an academic, is to lock down an SOP. We always know we can improve it.
It's very difficult.
Yeah, one last question I would have, Jeanne. One thing that you did point out is the funding that you received from CERM, a lot of that being local, and then the luck of having an advocacy team that wanted to come in and invest. I think, there are obviously other universities here that don't have the luxury of kind of local resources like that. Do you have any suggestions
I don't feel like the luxury,
for them?
This has been really hard fought. Even our first grants from CERM, they said, why on earth do you want to do gene expression? What does it have to do with stem cells? Every single one of those grants has been very hard to get through, and I haven't gotten them several times, because that idea of doing, it's just not, the study section, or the grants working group in that case, didn't appreciate it. So I think that, and at places like Harvard, I think, for example, we're all different. We're lucky because we're in California, we have access to CERN money. But there are other places that have access to money like that. I feel sorry for the people in the middle of the country that don't have other sources of income besides the NIH and, I don't know, whatever, So we're trying to do a distributed model in which we get funding from every possible source. So if we need to, we'll start a company and we'll get investment. But I don't want to do that until I really have to, because I don't want to have to go through the issues of having to license a lot of, I want to use the best technology. I don't want to use the one that I can license for the least money.
I also, one thing I found incredibly heartening about Jeanne's presentation was when you mentioned about the Parkinson's patients and the fact they understood the science. And I think for me, there's two historic bones of contention. One is that I think as researchers, we're wonderful at writing grant applications at a heady intellectual level, but then the people on the street who are actually paid for the research, if we don't do a job of conveying it to them, I think that's a little bit immoral. But the second part is a lot of grant-making bodies, particularly for late-stage translational grants, now have, for say, every 8 academics on the panel, 1 industrialist and 1 layperson.
Yeah.
And the layperson could be an expert in assessing the properties of drone regulation or something like that. But actually, unless we can convey it to everybody, then your grant could get held up.
I absolutely agree. I think communicating the importance and exactly how it works, showing that it's really very simple and straightforward, and engaging people so they feel like, yeah, that's the way I would have designed it, even though I'm not a scientist.
Yeah.
Questions?
Oh, yeah.
We have about 10 minutes. Are there any questions? Okay.
So with getting these technologies approvable in front of the FDA, there's really 3 stakeholders you have. I've got a whole new drug platform in front of FDA right now, and it's interesting to watch. I mean, a lot of these new expedited pathways and stuff are there so the FDA can actually make logical decisions about what to apply and what not to apply in terms of gating technologies. And I've been very enamored with those interactions with FDA. They follow the science very well. They're in an unenviable position. They're building the fire truck on the way to the fire, and have to make decisions on the fly. There are also obstructionist points of view that happen, and that mainly comes from this gigantic gaggle of consultants and advisors that practice in front of the FDA, who absolutely have no interest in having lower costs or less of their time, effort, and perspective involved. And they tend to hold you up and then really force the FDA into the mode of the other part of FDA, which is all the box checking. And there are many boxes that just don't have to be checked, in fact, make no sense to check with these new technologies. So the FDA science-driven part, great. The process suffers from all of this gigantic industry that grew up around it. As developers of new technologies and promoters of these expedited pathways, how do you handle this group?
So I guess everyone's looking at me there.
Yeah,
So I guess let me answer this from a very direct and I think pragmatic perspective, and then also as somebody who spends part of his life being one of those unfortunate consultants you speak of. And I think that from an academic viewpoint, it's always much easier with an academic hat to engage the FDA because you have no vested interest other than progressing things forward. And I will say that that's a blessing and a curse because, and contentiously, the quality of academic clinician-led trials is often far, far lower than industrial ones. And actually, if there was a way in which some basic principles could be applied, I think that you wouldn't have this end of phase 1, start of phase 2 cacophony of trying to work out, where the noise is. I think that the golden rule of regulation is if it wasn't written down, it didn't happen. And I think that all the box checking, as infuriating it is very, very important. I'd always be careful about speaking about the consultants for 2 reasons. One is that the majority of them are all ex-FDA or still sit on FDA committees, so you don't necessarily upset them too much. But also, it's fundamentally a communication issue, because you go from a process where it's the nomenclature is scientific and it's emotive, to something which is depersonalized and process-driven, and insofar as possible, standardized. And I think that is the big challenge. How can we take really exciting heterogeneous technologies that weren't really developed with the old regulatory paradigms in mind and have an open dialogue to try and make them fit the box as best as we can? And there'll be a few jagged edges, but how do we do that? I actually think on the consultant point, it's exactly the same as picking an academic PI for a trial. Make sure you pick somebody who understands both the regulatory pathway and the core underlying technology. Because what you don't want to do is have a scientific conversation in one direction and a regulatory one in another, where, as you said, they very much align. I also think that, Jeanne is available to assist with these submissions, should you so require.
Yeah, and I would actually say, Eric and I struggle where we really are required to work and partner with someone who's bringing something to clinic, because we really can't even have, as tool provider, conversations with the FDA that are too broad. We kind of need to understand exactly where it fits for them to make suggestions, and so it becomes even more important if you want new technologies in the market to really engage the tool providers to make sure that they understand what's being asked of them and are working within the compliance the FDA expects.
The other thing I'd say from a slightly, utilitarian viewpoint as a healthcare community, nothing frustrates me more than science being held back by bureaucracy. But actually something which frustrates me slightly more than that is things like, the theranostic situation, where actually I think it's done a net disservice to the industry. Because, I'm sure that in reality the detail isn't like this, but it's perceived in the press as kind of riding roughshod over what they'd already been told. And I think that just creates more uncertainty and more aversion to innovation, because then you go to investors and the first thing they say is, oh, are you another one of these? And I think actually we need to remember that the decisions we take today affect, our counterparts tomorrow.
Yeah, and I guess I would add to that too, that even when we talk about the level of data that you have from something like what Jeanne's working towards, that is what's really safeguarding that the patients are safe, so that we don't have things that could end up shutting down entire programs like the IPS programs or the new successes that are happening with gene therapies.
Yeah, yeah.
All right, I think a question right here.
Thank you for the insightful information. So regenerative medicine or stem cell therapeutics actually has become the horsepower for delivering the precision medicine to the world. Thank you for sharing with us this amazing results we've generated from the laboratory. However, we notice, surprisingly, the preservation method, which maintain the shelf life for all those live drugs, is severely behind. Like the technology we use today has not been really changed in the last 50 years. All the labs around the world has to deal with toxicity and deal with the circumstance generated from those solutions. So could you comment on that? And where do you see the future to standardize this preservation solution to allow effective delivery for the drugs? Thank you.
That was like Eric.
Yeah, I think that's an excellent question. I tried to touch on it a little bit in the presentation I gave, but you're absolutely right. The innovation in that space has not really progressed very much over the years, and I think that's partially because the requirement has been less commercial, right? So as we're moving these technologies forward now, the need for larger scale, better technologies in that space are going to drive that innovation. So there are a number of opportunities to improve upon this. I know that Thermo Fisher Scientific is certainly looking at opportunities. There are other companies out there that are highly specialized in the cryopreservation space. There are technologies with free radical scavengers, et cetera, that are used. But again, that's still pretty basic technologies. These are incremental improvements. Maybe you have an increase in viability post-thaw by a certain percent, or you have a thawing agent that helps the recovery of these cells. And that's great, it's one step, but the innovation and the focus in that area is right. There are a lot of opportunities for improvement, clearly. And the logistics of this, drones or no drones, is another big issue, right? I mean, ultra cold chain logistics is extremely expensive and inefficient. Biorepositories are very expensive to maintain. I mean, that's what we have today, and that's the state of the industry, but it's going to have to improve. So, I thank you for the comment. I don't have much of a suggestion at the moment, other than to certainly recognize that that's an important area for focus and innovation.
Comment too. So there are a lot of things I didn't talk about today. One of them is because we're cognizant of the fact that a surgeon is not going to be able to manipulate the cells. So we have to cryopreserve them. We have to figure out a way for them to thaw them and get them into a needle in a way that doesn't damage them. We don't want any thinking in the operating room at all. We want, So we've essentially tested the cryopreservation agents that are on the market. We focused on the GMP ones, and we found some that are extremely good. And the way we find them being extremely good is not just to look at the cells pre and post-thaw, but to put the post-thaw cells into animals, into our animal model, and ask whether they work just as well. So I thought that was going to be a much bigger problem. I'm still worried about how to make the surgeon do the right thing. But I don't think that freezing these cells, we can now freeze them at virtually every stage with really good results.
So not to dwell on cryopreservation, but I think just two dimensions to this problem. One is that we're trying to cryopreserve cell types which we never have before, and that's an inherent variability and a bit of process development. But the other thing is that actually, if you look in the industrial literature, we'll speak to people like Sartorius, Thermo, BioLife, etc. Cell banking of monoclonals, etc., which does require cryopreservation, has been around for years and is quite established. It's just that there's never been a need for researchers to have to bother before. So I think, reach out to your local charming reps, let Eric buy them a beer, and find out all you can.
Yeah, and I think, again, there's another new area for innovation, because you're now deciding, things that used to be put into vials, we're really moving into, they're in thawing bags, they're in some kind of a freezer bag that maybe didn't have 510K approval in the past, but now needs it. So this entire market is requiring a different level of regulation due to what the product is and how it's being handled in the hospital compared to the past therapeutics that are out there. I do remind everyone, Mark Zimmerman and I had a conversation about this yesterday, that 30 years ago monoclonals were in a different space as well than where they are, with a very fixed process and very well understood characterization of them. I think we're still in the early days, even though I think we'd all like to feel we're moving further along and things are moving quicker right now with more funding than we've seen in the past. We're still kind of in those early stages to getting to a point where it is a market and an industry that is more more bespoke and more standardized throughout.
Okay, great. We have time for one more question. This gentleman's raised his hand 5 times.
Thank you. So my question's of a scientific nature for Jeanne. First of all, thank you for a truly exemplary work in progress from stem cell to patient application. You presented early on in your talk the impressive results obtained using fetal transplants in some cases to treat Parkinson's. So in those cases where presumably there weren't very many serotonergic neurons,
Right.
but still a mix of cells,
Yeah.
I'm guessing it's because of the mix of cells that they did so well. Obviously, as you know, in the normal environment of these dopaminergic neurons, there are other cell types like vascular cells and support glia and other cell types. So I'm wondering, in comparison, what you might expect with your purified cell types, whether you'll get such long-term survival and improvement over time as the fetal ones, and if you've considered, I know this would complicate a lot the testing that you need to do, but including other cell types like vascular precursors, because we're seeing in mice that when we use mix of cells, we can have large transplant sites that survive very long and do very well because they're much more normal in their cellular compositions.
Yeah, so we've taken this has been a rather wandering path to the point where we are now, and early on I really thought that it would be very important for the cells to be with other cells that they appreciated. It turned out to be impractical for the point of view of evolution of our differentiation strategy, because it would be, we don't know what we want, so we had to try to get more dopaminergic neurons. Now I'm hoping that they don't require those cells because they'll get them from the environment where we transplant them, because obviously there are glia there that are going to be nice and supportive, and there's of course it'll be on the other side of the blood-brain barrier, so and there will be all these other cell types there. But the fact is, that would be a great thing to do when we decide to improve this process after we either, I hope, see success with these transplants, or unfortunately failure. That would be a really important thing to examine. I mean, my history in Parkinson's disease was is very old. I mean, I first did transplants in the late 80s. And at that point, I did I thought I'd done an extraordinary thing. I took cells from rat fetuses and I grew them in aggregates and I transplanted them and found that they all survived. Now, I would love to go back to that, but it just isn't practical. I mean, I would have to do it as a side project. So, yeah, I think it's something we keep in mind, but I don't see a way to actually test it in any meaningful way.
Well, thank you very much. That's all the time we have for this session. We're going to take a 15-minute break. So for everyone on the live stream, enjoy the videos. And thank you all so much.